All right, folks, let's get settled in. What I'm going to do today is wrap up our discussion on evolution of pathogens, and then we're going to migrate into something that I think is one of the cool topics of the class and helps, I think, connect a lot of the kind of molecular events that we talked about to the real world. So what do we actually kind of care about in terms of disease and managing diseases in populations. So before I wrap up, kind of talking about what are these like signatures that we see of evolution of pathogens, are there any questions from the last lecture? Jeez, guys, never any questions. Let me put. I guess the other item of business that I think is critically important. If you have not voted today, please carve out time to do so. Polls in Missouri are open until 7 p.m. Please. So class gets out at 5:15. If you haven't voted, get your polling place. Get in under the wire, and make sure your voice is heard. I don't remember what Illinois is. Is Illinois 7 p.m. too? I don't know if any of you are commuting in Illinois, but vote. All right. So I'm gonna wrap up thinking about the evolution of pathogens. So one of the things that we see, and this is going to be an important component of the rest of our discussion today and our final kind of informational discussion on Thursday. So this is the penultimate lecture from me in this course. So whether you're happy or sad about that, I don't know. But one of the things that is striking that we see when we actually go and isolate pathogens from infection events is that they're very clonal. And so what do I mean when I say something is clonal? It means that it essentially looks genetically identical, right? And so I spent the vast majority of the last lecture talking to you about how diversity arises and is selected upon in bacterial populations, and that this rapidly gives rise to very divergent subspecies of the same organism, particularly when you start mixing in horizontal gene transfer into the mix, right? So you have these the natural pace of evolutionary events that's happening in a bacterial population, and then you add in these sort of quantum leap events that are horizontal gene transfer events, and that these same processes are happening all the time. But despite that, what we actually see are very clonal outcomes in terms of when we go and find these pathogens in hospitals, for example. And the reason for that is that while there might be standing diversity in the population of a particular pathogen, is that the number of bacteria that are actually involved in initiating and being successful in a particular infection event in an individual, whether this be from eating some bad food, or that you work in a hospital, or you unfortunately have been hospitalized, is that it is a very strikingly small number of bacteria that initiate disease. And because of that, what you end up having are these very severe population bottlenecks that happen. So there might be a very large population of bacteria that you consume. So say you have 10,000 salmonella that's in a glass of contaminated water. It might only be just a very small subset of those salmonella from that population of 10,000 that actually cause disease in meat. And if I'm going to continue to spread this to others, this means that everyone else that I infect with this particular pathogen as well essentially ends up with the same nearly genetically identical version of the same pathogen, right? So the thing about this that's nice is this makes outbreaks traceable. And so this is why the Center for Disease Control and Prevention in Atlanta, why they care about isolating pathogens from specific food contamination events. You name it. Anytime that there are specific types of pathogens identified in hospitals, they're isolated, and those are sent to the CDC, where they're essentially undergo a battery of characteristic tests. So they're looking at what is the genome sequence of this particular isolate, what sort of toxins does it produce, all you name it. It's testing all of these things, because ultimately that makes these things traceable. So like placing a barcode on that particular version of the pathogen. And when you have enough information, you can engage in much more interesting types of questions if you're into epidemiology, and asking what are patterns of emergence of different versions of pathogens. So one of the things about this is that the ability to do this now is greatly enhanced by the ability to do next generation sequencing that I talked to you about before, so we can rapidly sequence whole genomes of pathogens and engage in a level of comparative biology that is just basically unprecedented. So this is a really, really exciting, interesting area of microbiology and disease-associated microbiology, medical microbiology. Being able to do this kind of molecular forensics of where did this pathogen come from, and our ability to essentially trace things back to sources and say, like, yeah, this farm was spraying cow poop on their lettuce, and that's why everyone got this particular type of E. coli from eating contaminated lettuce from this farm, because they're doing this. So I can't highlight this enough. But the things that really drive the evolution of pathogens are these selective processes that we talked about. But the horizontal gene transfer is one of the major vehicles that is driving the evolution of different types of pathogens from commensal E. coli, for example. So this is an example from E. coli. But there's this myriad of E. coli's that will infect different types of tissues, do different types of things, and cause different types of illnesses. Essentially, their ancestor was the same E. coli that benignly resides in all of our colons and makes our lives pleasant despite the horrible food that we eat. And that acquisition of different plasmids, ICE, DNA, lysogenic phages, all kinds of different pathogenicity islands that are encoded in different things. These things end up in E. coli through horizontal gene transfer. And those E. coli rapidly become specialized pathogens through very, very rapid acquisition events. Specific hardware basically to become a particular type of pathogenic E. coli. So, that said, acquisition events are one part of what happens to pathogens, but the flip side of the coin is that there's often gene loss. And so the example of Shigella that we've talked about a couple of times, that it began its life as a non-invasive E. coli. It just was a pleasant friend of us, and over its evolutionary history, it's become a very, very nasty pathogen. And so one of the first events that we know happened doing this sort of molecular kind of forensics is that it acquired its virulence plasmid. And we know that in part because we can take that virulence plasmid, put it into a commensal E. coli, and make that commensal E. coli essentially able to be it can begin to engage in invasive behaviors. So it can invade macrophages and epithelial cells. And that it has acquired a number of other things. But interestingly, along the way, it has lost a number of things too. And so some of these should be kind of interesting in that there are a number of outer membrane proteins that were lost early on in its history, and some very interesting ones. So flagella and fimbriae. So what selective pressure do you think drove loss of these things, given what we've talked about with Shigella? So Shigella is an intracellular pathogen, and it is a full-time pathogen. Like, that's what it does. It basically likes to spend its time interacting with us or other animal hosts. So what does flagellin or flagella activate?
The innate immune system.
The innate immune system, right? So if we kind of look at things that are lost, a lot of these outer membrane proteins, the components of LPS, these things have been lost through this organism's evolutionary time. And one of the arguments for these losses that we often see in these very specialized host-associated pathogens is that if you're under constant selection interacting with the host immune system, either you've acquired ways to combat elements of the host immune system, like many of you guys have seen with your specific pathogens, or you just give up those things entirely, right? And interestingly, loss of sort of generalized metabolism is a feature that we often see. So if you don't ever see particular substrates in your life anymore, if you're a bacteria, you give up those particular pathways. And that there are modest fitness increases for giving up catabolic diversity. So metabolism versus anabolism. Metabolism is essentially what you're using to burn energy versus what you're doing to make it, or to make biomass. So catabolic pathways, like losing them. So Shigella doesn't eat lactose, E. coli does. Shigella does not eat ribose, E. coli does. There's a number of other catabolic pathways that Shigella does not have, so it will not eat a lot of different carbon sources. It doesn't produce a large number of secondary metabolites, a whole bunch of things that it doesn't do as a result of basically these losses being selectively beneficial for Shigella. And last is that why do these things happen, and why do we see acquisition, loss? Why do we see these sort of radiations of different pathogens? In part, there are these constant population bottlenecks happening in hosts, and in addition, that we spent a good amount of time in this course thinking about this interaction with the host. This is a major selective event for pathogens, and indeed it is, right? And so for some pathogens that you guys are looking at, if they have a life that they lead with one host, and that their sort of zoonotic interaction with us is a very bad one, right? So there are some pathogens, for example, that live in chickens. They're commensal to chickens, and they are a terrible, terrible pathogen to us. And where they spend a lot of their time, if you are not an obligate, host-associated pathogen, is that they're out in the world, right? And so all of these horizontal gene transfer events, arguably very few of them happen in us, and where they really happen is out when they're not associated with us or associated with the host. So when they're out living, if you're Vibrio cholera, you're living on like the sort of decaying remains of phytoplankton, for example. And that is where you are sort of engaging in transfer of DNA, acquisition of phages, all sorts of things And then essentially these populations come into contact with us, undergo intense hard selection, and we end up with this thing happening over and over and over again. And one of the few sort of breaks from this that we see are these, what we refer to as like niche restricted adaptation. And so essentially you become an obligate pathogen that you absolutely require your host for your entire life. And so you do not live out in the world, out in this genetic melting pot, that you are hardwired to live in this one particular environment. And so some of you guys have some interesting examples of these niche-restricted pathogens, but many of the pathogens that we've talked about in this course are essentially cycling through this never-ending, iteration of being out in the world, acquiring new things, losing things, coming in contact with host organisms, being subject to intense selective pressure for specific traits, and this cycle continues and continues and continues. So one of the big questions that's out there now is, given the amount of information that we have, can we start to predict what's going to happen with some of these events, right? So one of the things that you're always doing when you're amassing data, large amounts of data, is you don't do this just for the sake of doing it. You want to do it to ask a question or to do something predictive. That's one of the missions for CDC, for example. How do we predict what is going to happen to pathogens as they sort of iterate through this never-ending cycle, and what should we be anticipating happening next? Things like antibiotic resistance, acquisition of toxins into pathogen lineages for pathogens, you name it. So what are we looking at on the horizon for what's going to kill us next? So, are there questions? So everyone's really cool if I ask like a really hard exam question about this. Let's do it. Yeah, second? Okay. For those of you interested in these sorts of topics, I hope I've kind of highlighted some exciting areas to be thinking about. So, where are we going next? The next is one of my favorite topics in this course, in part because it's relatively quantitative, which unfortunately generally means that lots of people don't like it because there's people. And the reason that I want to drag you through some math that I'm going to take you through today when we're talking about epidemiology is that I think it's really informative to see where the basics of some ideas come from, and give you sort of a framework to understand when much more complicated epidemiological models are developed. They all begin somewhere, and I'm going to kind of give you the basics of where this comes from today. So, things that I want to address today related to epidemiology, and we'll see how far we get today. I at least want to get through this first block. And these questions, why I'm going to leave the lights on is because I'm going to take you through some things on the board, is how does this relate to bacterial pathogenesis? And I think this kind of comes full circle to one of our first lectures, that really the topic of bacterial pathogenesis is one of the few places that we have enough data related to molecular events that we can begin to make connections to much more kind of global scale events and why a particular pathogen is so successful. I'm going to give you some info on what is this idea of a basic reproduction rate, or basic reproductive number, and then some ideas of where does this come into play when we think about disease control and prevention. And then if we have time, I'll kind of take you through some of these ideas of what's going on with emerging infectious diseases versus re-emerging infectious diseases, and that's something else that I touched on before. So, according to the World Health Organization, Epidemiology is the study of the distribution and determinants of health-related states of events, including disease, and the application of this study to the control of diseases and other health problems. Okay, so long-winded, it's sort of everything, right? And really, if I were to distill this to what I think this means, is that you are trying to understand the transmission of diseases in populations, and ultimately connecting, how successful a particular pathogen is, relating it to its particular phenotypic traits that it has. And so how does this apply to bacterial pathogenesis? Well, about 25% of deaths in the world are caused by infectious diseases. And if we were to kind of look at sort of the major players in killing people, the large majority of these things are caused by bacteria. This accounts for the majority of deaths associated with infectious disease. And so why our Center for Disease Control and Prevention spends millions and millions of dollars a year surveying and tracking bacterial pathogens is because they're one of the chief mechanisms for our demise. And so, what does epidemiology have to do with pathogenesis? So really what epidemiology is helping us do is connect this disease phenotype and exposure, right? And sort of drawing a big black box around all of these molecular mechanisms for the most part, and saying that those processes happen, but how is the disease connected to its transmission, and how are people exposed to this particular pathogen? And so you can sort of calibrate these sort of epidemiological models with ideas about how nasty is this pathogen, and how deadly is it, how quickly does it spread in any particular host, and how quickly do we see manifestation of disease, and what does that mean for exposure to more people? We'll get to some of that. So, epidemiological studies, they do not explicitly require molecular details. So that's one of the nice parts of epidemiology, but they are deeply informed by these details that we've talked about. So if you're saying, we've done particular types of epidemiological models for Salmonella A versus Salmonella B, we see Salmonella B is way worse for everybody than Salmonella A. If your next question isn't why, then I don't know what to do. So why is sort of the next question that gets immediately precipitated in those sorts of observations, and that's where all the details that we've spent kind of ages talking about in this course come to play. And so understanding why certain individuals are more susceptible than others, and how the pace of disease onset and duration are controlled by aspects of this host-pathogen interaction. So why Salmonella typhi is so much worse for everyone in this room than Salmonella typhimurium, right? And it all comes down to the details. And if we actually want to be much more careful in sort of building predictive models for epidemiological studies, these parameters that we're going to talk about in a very simple model, they can be deeply informed by what we understand at a very, very low level in terms of kind of the nitty-gritty details of a particular pathogen can help us calibrate these models and make them much more clear. All right. Any questions so far? So, if I were to throw some vocabulary at you in the next couple slides, there's a lot of terminology you will see related to epidemiology, so whether things are a chronic or acute infection. And so this is this idea of, is this a long-term or short-term infection in a host? So some of the pathogens you guys are looking at are very acute pathogens. You're going to have a nasty infection, and you are going to clear that thing, or you're going to die. And others have examples of what we term as very chronic, long-term infections in host. So this relationship is established, and you're going to, if you are infected with one of these pathogens, then this is going to be a long-term, nasty relationship. And who's heard the word epidemic? Who's heard the word pandemic? So what this refers to is that epidemics are local increases in rates of disease occurrence. And so I might say, and it all depends on perspective, right? So if our perspective is the UMSL campus, and that's our entire world, an epidemic would be this room or this building having an outbreak of something. And a pandemic is referring to a region or international increased rate of occurrence. So essentially, are we seeing that not only this Benton Hall, but all of the science complex and all of North Campus have an increased rate of salmonella or something? And if we take a much more global perspective, this is where lines get blurred, right? Are we talking about a state-level epidemic, and is it a pandemic if we're talking about everywhere in the United States? It's I would argue that it gets murky. And so the most kind of classic example in sort of delineating these two things are, if you were to think about influenza, it's not a bacterial pathogen, but some of the same principles, like a lot of the same principles apply. And we'd be talking about an epidemic in the United States, and we'd be talking about a pandemic if we're talking about worldwide increases of occurrence of influenza. And there's mortality. And so this is essentially the death caused by this particular pathogen or thing in a population, which is in stark contrast from morbidity. Okay? And so morbidity are essentially the incidence of disease in the population accounting for fatal and non-fatal outcomes. And so basically, that this is kind of everybody that's getting sick versus how many people are actually killed by this particular pathogen. All right. And so this all comes back to there are aspects of bacterial diseases that an epidemiologist is going to have some different language for than what we've talked about so far. And essentially this is drawing this black box around all of the molecular events that we've talked about. And so there's the infection event, and this is essentially you being exposed, acquiring this pathogen. This organism invades, colonizes, and grows in the host. There's this incubation period, and essentially the time required for onset of disease symptoms. So this acute period, the height of disease symptoms in the host, decline period, where essentially you're getting better, and this convalescent period where a patient recovers to normal health. And so I don't know about anyone here, but if you ever had food poisoning, it's not like 48 hours, you're like, well, I can now leave the bathroom. And so now you're maybe between 4 and 5. But number 5, that's a long time, right? I'm not ready to head back out and go to my favorite restaurant and get a giant dinner after food poisoning, right? So the convalescent period. So this approach back to normal health, depending on the severity of what this disease associated with pathogen is, you might never get back to normal, right? So this might be a lifelong trajectory back towards normal, and it might be lifelong. So hopefully it's disease. All right, so any questions so far? And so if we're thinking about epidemiological events, and so thinking about sort of population level disease transmission, there are sort of two bookends to what you would imagine in terms of how infectious diseases are spread. So you have direct host-to-host transfer, so this requires requires direct contact, touching, etc. And this can also be indirect in that me being close to this table, and if I were to have a horrible hacking cough and not cover my mouth, I could potentially get everyone at this table sick, right? And so that falls into this category of direct host-to-host transfer. And there's also indirect host-to-host transfer, and this is where you're really requiring vectors. And so whether these be live vectors, things like insects or other vertebrates, that are transmitting something, so they're a carrier. They can be a neutral carrier. They can be also a pathogen, like a host for this pathogen in terms of they're making these animals sick. And there's also inanimate carriers, so sort of like hospital beds, instruments, food, all kinds of things that are sort of these middle intermediaries between hosts. So which then begs the question of, well, what does this mean for epidemics? And what this means is that you can have sort of bookending things again. You can have common source epidemics. Essentially, there is one source for all of these infections. Or you can have host-to-host epidemics where you have a couple of infection events, and they infect a couple more people, and they infect some more people, and they infect some more people. And where these really become the signatures for these in terms of if you're doing surveillance, where this really comes to play is that it really comes down to time, right? And so if you're closely monitoring things in terms of time scale, this gets back to what we talked about in terms of thinking about you actually want to monitor the evolution of something and sort of understand what sort of different selective pressures are there, what types of beneficial mutations are out there. It really requires having appropriate resolution in terms of time in your sampling. The same is true here. And so if I were to measure at time 1, and I were to see, well, there's 4 people that all have, in this room all have salmonella. In both cases, it would look the same. Now, if I were to monitor every single day for 2 weeks, and then all of a sudden, today I see, wow, there was 4 people that suddenly got salmonella poisoning, but I might be able to say, all right, this is a common source epidemic. So, where did you guys all go out to eat? Oh, we all went to our favorite restaurant and we all got sick, right? Or the contrasting event is that, all right, I was monitoring every day, and last Friday I saw 2 of you had salmonella food poisoning, and then today now I see 4 people, right, that have this happen. And where you're left as an epidemiologist trying to understand what in sort of real data these sorts of things happening, this is where you require information from people, right? And where did you go to? Where have you eaten for the last 7 days? Where have you done X for the last 7 days? What where all this sort of meta information is important in understanding and delineating between common source and host-to-host epidemics. And so I'm giving you sort of the bookends, but these are the extremes, right? And in reality, many outbreaks of particular pathogens are somewhere in this middling gray area in between these two broad patterns. Right? So are there any questions so far? No? So now, here's where some math begins. And so one of the really important numbers that an epidemiologist cares about is this basic reproductive number, or R0, R0. And what is this thing? And what this is, in very straightforward language, is it's the expected secondary transmissions from each single case of disease in an entirely susceptible population. So if someone walked in that door with cholera, the question is, if they were in here for an hour, how many people would you expect to get cholera? Right? And so this is what this number is helping us get a handle on. And so it's a mathematical construct used to parameterize and quantify disease spread. Okay? And so it is a unitless number, so it's not a rate. It's not something per hour. And even though it is sometimes referred to as the basic reproductive rate in some literature, I like to say it's a reproductive number, because units do matter. And very simply put, if R0 is greater than 1, this disease will spread from one infected individual to more than one susceptible host, right? So we would expect that this thing is going to spread quickly, and if R0 is less than 1, an infected individual does not typically transfer disease to a susceptible host, right? So this does not mean that an infected individual can't wreak all kinds of havoc. If I had cholera, because if I had cholera and we were all drinking water from the same well, and I hadn't washed my hands, and I washed my hands in the well, and we all go and get water, we would end up with a common source epidemic, right? And so, but if I crunched all the numbers in terms of parameterizing things with cholera, its R0 is pretty low, right? And so what these R0 values can tell us as well is if we know how a particular pathogen likes to spread through susceptible host populations, it can help us begin to delineate whether or not, like, yeah, this is totally a common source epidemic, right? Because if I know the R0 for Vibrio cholera is pretty low, and then I just saw a massive number of people get cholera, I would expect that it is probably a common source epidemic, right? Because this R0 value is telling us really how well does this host-to-host transfer happen, right? I'm infected, I infected 2 people. Those 2 people infect 2 people, and so on and so on and so on. Versus, I showed up, I'm sick, I contaminated something common to everyone in the room, and everyone in the room got sick, right? So if we break down how you would calculate this, is you would need to know how many infections happen per contact, how many contacts happen for some unit of time, and what is the time per infection, right? And so the constellation of taking these variables and sort of parameterizing them into tau, C, and D, you can sort of say, all right, if the infections per contact are low, but the time per infection is very high, I can ascertain that, an infected individual is going to be infected for a really long time, and while the infection per contact, it's an inefficient pathogen in that it doesn't infect everyone that I come into contact with. Throughout the course of my infection time, I'm going to come into contact with a lot of people, right? So maybe the time per infection is really, really low. And so I'm sick for a day, but anyone that I come into contact with is going to get this, right? So there's a lot of different ways that you can have an R0 be very high. And there are a lot of ways that you can have it be very low, even though some of these things might be individually very high, because essentially it's a product of all of these values. So you're multiplying all of these values together, and if it is greater than 1, that means this pathogen is prone to spreading through a population, and if it's less than 1, it tends to not rapidly spread through a population, right? So that's nothing else. Greater than 1, less than 1. What do those two things mean? So where does this entire idea come from? And this is where we're going to do a little bit of math. So we have a population of people, or animals, plants, whatever you want, and it has a fixed size n. So there are n individuals in this population. And so rates of transmission, removal, etc. are constant. So the number of, and that there are no births and deaths, and that it's well mixed. And so everybody's moving around the room. Nobody new is coming in the room. No one's leaving. And essentially, we have a fixed population size. So in our population of n people, let's assume there will be a fraction of susceptible people. And I'll just draw our population of susceptible people. One more. Two more. Okay. All right, someone wrote it black, so this one probably works. Okay, so there are 3 populations of people. We're going to have susceptible people. We're going to have infected people, and we'll ultimately have resistant people. And so how do you think you become resistant to most pathogens?
Been exposed to them before?
You've seen them before, so you unfortunately had to be one of these folks, right? And so you can move from the pen of infected people into the pen filled with resistant people, and that's great, right? Now, susceptible people, unfortunately, you get infected, right? Now, the thing that's simple and nice about this particular model is that no one dies, right? It's the simplest possible model we can have. Now, the thing that's important is that each of these groups of individuals, if something is happening in terms of what's happening to these different populations of people, is that these numbers are going to change through time, right? So the number of we would anticipate that the number of infected people is going to increase if susceptible people are exposed to some pathogen, and that a number of infected people who don't die will eventually become resistant because they've already experienced this pathogen. So we can make some very simple relationships about this and saying the rate of change of something is always going to be equal to the amount of something coming in minus what's going out, right? So this is just a very simple mass balance. If we were to draw a box around every one of our groups here, let's say there was S people, there's one arrow going up, right? So susceptible people become infected people. So there's no inputs into susceptible, right? There's a population of initially susceptible people, and that we only see output from that group. So there's only people leaving that group to become infected, right? Similarly, if I were to draw this same box around my infected population, that here's my input, here's my output, that the input is essentially everyone that was a susceptible person is becoming an infected person. That's the input into this population. The output are the individuals that are infected, becoming resistant. And finally, our last group of people are these resistant folks. And that there, again, there's no output, that you become resistant. And then in the simplest incarnation of this model, that no resistant people become susceptible. So you're resistant for life to this particular pathogen. And so we only see inputs into the resistant population, right? And so we can think about all of these things in terms of their rate of change in time by saying, well, what is the change in the number of S per change in time, right? And I can do that for all of these. But now, if I were to say, I want to take the limit of this, because essentially thinking about things in sort of discrete variables doesn't really lend itself to doing some straightforward math. So I'm going to employ a little bit of calculus here and say, I'm going to take the limit of this as the time step of what I'm measuring goes to 0. And what this ends up doing is saying that, well, now we're going to say that we have the change in the number of susceptible people over the change in time. And essentially, this is the first derivative with respect to time for the number of susceptible people. And I can do the same thing here. I can say this, and I can say this. Right? So, to save you some pain with bad handwriting, is we can set up a fancy set of this differential equations. And what am I getting at here is that essentially we have, if we go back and look at this, is that so tau times c, essentially that is a rate constant. And that if we assume that an infected person and a susceptible person come together to make two infected people, what we see leaving our susceptible population are things happening at that particular rate. That they happen in a second order fashion where the concentration, the number of susceptible individuals and the number of infected individuals have an important bearing on the rate of change of susceptible people in our fixed size population. Similarly, what you should notice is that essentially this output term here, which is the rate at which things are leaving here, is exactly the same as what's going in to our infected population. Similarly, the things leaving the infected population are identical to what's coming into this resistant population. And so what I've set up here is a system of differential equations. So I don't know if any of you guys have seen this in calculus, but it gets easier in just a second. So the idea here is essentially we're just accounting for what's happening, what's moving in out of one population into another, right? And all of these numbers, or all of these things like tau, c, d, all these things have to do with sort of measurable parameters that we can say about how fast something is happening. So, you know, how many infections happen per contact? How quick do these contact times for infection happen? And that can tell us something about, all right, that sort of calibrates how quickly these reactions between a susceptible person and an infected person happen to move from a susceptible person into being an infected person. Okay? And so what's very similar about this is essentially chemical kinetics, right? The same idea is going on for, if I were to say, you know, I want to model a set of chemical reactions. The same concepts are alive and well. Okay, so what would need to be true for an epidemic, right? And the simplest interpretation of an epidemic is that the number of infected people have to be increasing, right? And so if that is true, that means that this DIDT, the rate of change of infected people, has to be increasing, okay? And so what this then means is that this, the IDP, the rate of change of infected people, that has to be greater than 0. So what I can do is I can say, all right, well, if this needs to be greater than 0, I can plug that relationship in, and all of a sudden move things around. This here has to be greater than this term, right? So the things coming in have to be greater than the things going out, right? So if that is true, you end up with that relationship there. And if I move things around, divide by I, move things to the other side, I can end up with tau CSD is greater than 1, right? So where does this R naught come from? There's one last assumption that's made, is that if we say that everyone in the population is susceptible initially, we can say that S is approximately 1 or 100%, right? So if S is very close to 1, which would be true when an outbreak is about to happen, essentially we can say this is equal to 1. We end up with this, right? So we've seen this before. And we just basically call that constellation of variables R naught. Okay? So the big tricks in all of this are starting with something that is a complicated nonlinear system of differential equations to describe a very simple process, and through making one simple assumption, and that is that, all right, the number of infected people have to be increasing in this population. We can use one of those equations from our system of differential equations to calculate something useful, right? And so this R naught value has to be greater than 1 for this pathogen to be spreading in a population. And so where did that all come from? It was from this very simple model that is the SIR model in epidemiology. So the susceptible, infected, resistant population model. Okay? So before we move on, what might you do to that model to make it a little more realistic? Is everybody resistant forever?
No, because the older you get, the lower your immunity is. Just so elderly people are more.
Right? So what I could do to make this a little more realistic, I could say, all right, now I'm going to draw another variable. Now, people in this resistant population can become susceptible. You guys think population sizes are constant?
No.
No, right? Do people die in diseases?
Yes.
All right. So now, if we say, not only is there infected and you become resistant, what if we have a population of people that die, right? So that changes this model. And so you keep adding things onto this very basic background, which arguably is pretty complicated in and of itself, to essentially make more realistic, more predictive models of how diseases behave in populations, right? But one of the things that using this really sort of bare bones model lets us do is just essentially come down to brass tacks, what has to be true for an infectious disease or a pathogen to be a good pathogen in terms of it's spreading through host-to-host contact. And that means that this basic reproductive number has to be greater than 1. Okay? Now, where does this come to bear and where is it important? Where it is most important is this idea of herd immunity. Who's heard of this term before? In what context?
Vaccination.
Vaccination, right? And so what vaccination has everything to do with is the size of this resistant population, okay? And so one of your homework problems is to derive this equation, to make a couple of different assumptions about what we talked about before. So if the proportion of immune individuals is high, there are fewer susceptible individuals, correct? So if you're resistant to a pathogen, you've gone through that horrible daisy chain of getting sick and then you're fine, right? And if the susceptible fraction S is much less than 1, it will make transmission more difficult, forcing the pathogen to extinction, right? So if we go back here, is that if S becomes very, very small, then this entire first term of this, what goes into that infected population, becomes very small. And so the rate of that happening goes down. If it's sufficiently low, it can mean that the other side of this equation is much larger than the first, which means that now the number of infected people is going down. The rate of that happening is negative, right? So what we can do, making a couple other assumptions that I asked you to do in the homework, is we could derive another very simple relationship known as the herd immunity threshold, which is equal to 1 minus 1 over R naught. What this helps us calculate is the threshold or fraction of the population that must be immunized and be put into, basically artificially put into that resistant group, bypassing ever being infected, to essentially force this pathogen into a situation that it doesn't have sufficient susceptible hosts to transmit through a population, right? And so if we were to plot out this HIT versus R naught, we can basically get this nice curve. And so what this nice curve lets us do is I can go into the field and I can study my favorite pathogen and work out, based on surveillance data, what the R naught of that pathogen is. So there's some standard frameworks to do that. The experimental frameworks that I can work out what that R naught is. It could be from E. coli, it could be Salmonella, it could be cholera, it could be Bordetella, pertussis, it could be influenza, it could be Ebola. It doesn't really matter in terms of conceptually, like this idea of becoming susceptible, to becoming infected, to becoming resistant to a particular pathogen. This is irrespective of whether it's a bacterial pathogen, whether it's a viral pathogen, or whether it's a virus. Conceptually, it's the same ideas. Here we care about bacteria. And what we would need to know is we can work out from other epidemiological data what R naught is. And the nice thing of why that model, that very simple model, is so useful is that it's a wonderful conservative estimate to say, you know what? I worked out that R naught was about 5 for my pathogen. I'm going to go up here and say, all right, about 80% of my population needs to be resistant, right? And so if I'm going to target that number, the best way that we have to generate a large resistant population in a population is immunization. So vaccination, right? We'll talk about that next time. But this also speaks to why vaccination is so important, right? That how many there are, if we were to incorporate death into a model like this, is that there are a good number of people in any population that cannot be immunized, right? And so this whole idea of herd immunity is that this resistant population in a population size of N is sufficiently large that this pathogen can't spread and basically kill off all susceptible people in the population, right? And so I don't know about people in this room, but I care. I do love my neighbors. I like all of you. And I want you to stay healthy, right? So one of the great tools that we have in terms of everything that I've taught you in this class is our ability to essentially artificially move people from being susceptible to being resistant if you're otherwise healthy, without having to have migrated through being an infected individual, which in many cases is terrible, right? And we'll see many examples of this with many of the pathogens that you guys chose as talking. So the trick here, and the rough part is that the higher this R naught, the more people you have to immunize, right? So as this number gets larger and larger and larger, and this second side of this equation gets smaller and smaller and smaller, that means you have to immunize more and more and more people. And so, what does this mean in terms of pathogens? So what I'm showing you here, and I encourage you to kind of zoom in on this slide because there's a lot of interesting data here, is that this is some data from the CDC and WHO, in that their epidemiologists have, through the years, gone through and worked out various R naughts associated with a wide variety of bacterial and viral diseases, and essentially what is their fatality rate. So what is the percentage of people that are killed if they're infected with this particular bacterial pathogen or virus? And so what you can see is that a lot of things are kind of clustered down here. So they're not particularly deadly. I mean, I wouldn't want any of these things. But their R naughts are really variable. And so, how do you think you guys would make some decisions about developing vaccines? So what on this graph, what ordinates and abscissa parts would you be looking to target? High
mortality rate.
High mortality rate, right? So if this stuff is going to kill you, it's awful. Are those things that you want to try and develop vaccines for? Darn right you do. So how about rabies up there? So its R naught is really low, right? So it's kind of barely over 1. The incidence of transfer from individual to individual is low, but if you do get rabies, it is bad news for you, right? So we care about that. We care about that because that fatality rate is like 100%. Like you're going to die if you get rabies and it's not treated, right? So yeah. So how about HIV? Sure. We'd love to have a vaccine for HIV, right? And that's why we spend millions and millions and millions of dollars, arguably billions of dollars, trying to do that, right? But this is where things become recalcitrant to actually making effective vaccines. That making vaccines ain't easy, right? It's not trivial to do it. How about TB?
Yes.
Mortality is pretty high. Like R naught's pretty darn high. It kills tons of people every year. It's one of the most nasty infectious diseases out there. Yeah, you want to make vaccines for that. So the things that I've talked to you a lot about in this class, so Salmonella, E. coli. Do you think we have vaccines against these things? No, right? They don't really kill a lot of people. They can. They don't in general. They're self-clearing pathogens. There are not, there's around one. Like, they sort of stably maintain themselves in populations, but they're not killing massive numbers of people. And so there's not a lot of push for a salmonella vaccine, right? Now, things we do have vaccines for. So rabies is one. So way out here with these really, really highly contagious infectious diseases. So whooping cough is caused by, is caused by Bordetella pertussis, and that is a particularly nasty infectious disease. You know, it's one of these like, untreated, it's going to kill like 5% of typically children. And so we care about these things because they're highly infectious and they have a essentially preference in their fatality rates for affecting vulnerable populations like children, right? So we care about things like whooping cough. We care about things like mumps, measles, malaria, diphtheria, all sorts of things that aren't necessarily terribly fatal, but they have drastic impacts on people's lives. And like, these things are really, really nasty infectious diseases. So, the other one, if anyone has ever had this, is norovirus. Has anyone had norovirus? It's like a cruise ship bug. It is awful. It is like top 10 worst experiences of your life if you get it. So it's basically 48 hours of horror, right? And so I do not wish this on anyone, but there's actually a lot of money put towards developing a vaccine for this. In part because it is 2 days of absolute terror. And where this typically happens is on cruise ships, right? So there are companies that operate cruise ships that pump money into saying like, hey, can we do something to get rid of this? So currently, the answer, I believe, is no. But this is why I won't go on the cruise ship. So anyway, I think this is one of the like really exciting plots to actually sit and look at when you're starting to think about like, why do we care about certain diseases and why do we develop vaccines for certain things and not others, right? In part, it has to do with really how rapidly is this going to spread through a population and how quickly is it going to kill people, right? And so those two variables leave us with saying, all right, if we're going to put our money anywhere, we're going to put our money on things that we can curtail really quickly by implementing vaccination schedules using that relationship. So, are there any questions so far? Who likes math? Well, this is a science class. Science rooted in math. So, where I'll end today's discussion is on these ideas of emerging and re-emerging infectious disease. And so, this is something I talked to you very early on about, and one is this idea that emerging infectious diseases, they're appearing for the first time. And, or they existed previously, but we didn't know that there was a particular pathogen associated with that disease. And so how often do you think these happen? Either rare or common?
Lately, a lot more common. people not vaccinated in their kids, so it's not
So emerging or re-emerging?
reemerging.
So re-emerging, certainly. So it existed previously, but this incident or range is rapidly increasing. So you start sort of playing games with this model that we've sort of engineered in our favor in dense human populations. You know, we see re-emerging infectious diseases, but emerging diseases they're pretty rare. And so things that really shape this, we've seen this slide before, are evolution of bacteria. So all of these events that we talked about last time. Human behavior, right? So making selective choices about your lifestyle, about lifestyle of those associated with you, vaccinating or not vaccinating, for example, and your environment, right? So if we have 40 people in this room, or if I stack 200 people in this room, those are two very different environments, right? And so as human populations change, undoubtedly all three of these things are deeply intertwined, right? Sort of isolating these as sort of pure variables, I don't think you can really do that, but they're sort of discrete factors. And so how do pathogens emerge? So one mechanism is zoonosis, and so this is where essentially a pathogen undergoes host switching. So perhaps a bacterial pathogen has been associated with chickens forever, and we raise and process lots and lots of chickens in the U.S. So in many ways we sort of manufacture an environment that promotes zoonosis, right? So if you have relationships in terms of like proximity to animals, to all sorts of other things, being exposed to something that's not normally associated with humans, now that is a human pathogen through exposure. You can have human-made environments and materials that foster increased exposure. So medical implants is a huge one. So Staph aureus, I know there's a group talking about it, for example, medical implants. Throughout human history, Staph aureus has been associated with us for a long time. And sort of deep tissue opportunistic events, for Staph aureus in human history, we generally probably revolve around I stepped on something really sharp, I was stabbed with something really sharp. Now we essentially, I'm going to open you up and replace your hip, right? And so that is something that didn't happen. That's something very, very new. So Staph aureus being the type of systemic pathogen that it is now is in some ways in part because of things like implants, air conditioning systems, water purification delivery systems, all these sort of like human-made things, human-made environments foster the growth of bacteria that would never be associated with at the titer that they are in these environments, or that we discovered diseases are associated with particular microorganisms. So I've mentioned this story about E. coli 0157H7. So it's a she-goat producing E. coli, loves to live in cows, doesn't do anything to cows. Commensal lives in the hind ends of cows. But due to high throughput meat processing that really kind of blew up in the post-Second World War era of food production, this pathogen is essentially sort of jumped from cows to being now a human pathogen. And that all of these things that it was normally able to do, it had all the hardware to make us sick. It just didn't do it to cows. It does it to us. So we've increased the frequency of exposure to this particular pathogen through food production practices. One of the classic stories, I'm not going to go super into detail about it because there's a group talking about it, Legionella pneumophila. This particular pathogen invades alveolar macrophages, loves to live in air conditioners, as the cause of Legionnaire's disease. So when artificial air conditioning was introduced to wherever the French legionnaires went and were occupying places, this is something that would throw in these old water-cooled air conditioners and essentially get people sick with a respiratory infection. And it loves to invade macrophages, but its ancestral host are amoebas. It turns out amoebas behave an awful lot like macrophages, and so it was able to make this really quick host switch, interacting with humans instead of just amoebas. And last are this interesting pathogen, Porphyromonas gingivalis. And so this was an emergent pathogen in my lifetime. And so periodontitis, this is something that was associated with basically like, well, you're not taking care of your teeth. Yeah, there's some bacteria that grow in your teeth. I don't know. Like, maybe it's caused by that. But due to advanced culturing methods, they were essentially able to isolate this particular pathogen and directly associate it with tooth decay, right? And so this is the critter that is nasty and causes really nasty periodontitis, so essentially chronic inflammation, tissue damage, bone loss, cavities, all the really nasty stuff that can happen to your teeth because of this. So, again, re-emerging changes in human behavior, evolution of a pathogen in response to the host, and evolution of antibiotic resistance. And I don't know if we're going to get to that in this course. But again, vaccinate, 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 and vote, vote, vote. Okay? So, see you guys later.